I'll let the rest of you take your seats, but just while you are doing that, my name is Selina Stead. I'm the CEO of AIMS, the Australian Institute of Marine Science, and I'm absolutely thrilled to do this introduction for our next plenary. I would like to start by acknowledging the traditional owners' lands on where we meet today and pay my respects to elders past, present, and future of all those on where AIMS works. I'm going to go off script. Most people know I will often do that, because I really do believe that Cynthia Reginas really needs no introduction. She is one of our AIMS rock stars, if I may use that. We have lots of rock stars, some a little bit smaller, different stages of their careers, but I'm really thrilled not just to be able to showcase some of the fantastic research that Cynthia has been leading, and which has also inspired quite a number of the talks that have been here at the conference, but also because the first conference I met Cynthia at, which was at Newcastle University, the Reef UK, I wasn't actually allowed to say that we would soon be colleagues. So it really is. I'd just been appointed as CEO, so it really is thrilled to be here. But very briefly, Cynthia did her PhD 2000 at the University of Arizona, did a postdoc at Duke University, and has been at the University of Queensland since 2006, becoming a professor very shortly after that. And since 2024, she's been the research team leader for our coral reef adaptation program and is our senior principal research scientist. She has so many wide-ranging expertise, I couldn't possibly do justice. So with that, I will hand over to Cynthia, and please join me in a round of applause to bring her on.
Thanks very much, Selina, for that really lovely introduction. I'm very humbled to hear all those nice things said. And I also wanted to really thank the organizing committee, and particularly Matt Nitschke. It's an absolute privilege to be here today speaking to all of you. I also need to thank the co-authors on this talk, the ICRS, who produced the data that will go into what I'm going to talk about today, as well as their supervisors. These are wonderful young scientists who teach me new things every day, and it's an absolute privilege to work with them. I would also be remiss not to acknowledge the traditional owners on the land we're meeting today, and the many sea countries on which all the people in this room are doing work. It would be a vast number. And I would like to pay my respects to elders past, present, and future. So when we look at the news and see things like this, I'm going to assume and hazard a guess that everybody in this room is fairly appalled by what we see and feels great anxiety over this impending disaster that's headed our way. And of course, addressing the root causes of climate change is the absolute most important thing. But even if some geopolitical miracle happens in the next year, climate change is baked in for the next couple of decades, and that's just physics. So the hot weather is coming, and we need to act accordingly. Now, keeping that momentum of a warming world in mind, all the scenarios for the future that show some glimmers of hope have one thing that a common thread, which is that evolutionary adaptation or genetic change is part of that predicted outcome. So adaptation can make a better future for corals. And I just stuck up a couple papers here, but there are many more. Okay, so how do we incorporate adaptation in our work? And in sort of more generally, how do we incorporate science in achieving an outcome of healthy coral reefs, right? So we have the end point, and there's wonderful managers that are here that are doing amazing things to make our reefs better. But as a scientist, I sort of sit on the other end. And so there's this gap between how do we get from the science that we do to real world outcomes. And particularly when we think about how science is really done, we tend to create anecdotes. We focus on single species, single ecosystems, et cetera. So how do you sort of add those all up? And we have this vision that somehow seamlessly our work is going to translate into something that is meaningful in terms of making the world a better place. And I guess the way I see this translation as happening is, can our work come to some general principles? Can we derive general principles that then allow managers to make informed decisions, right? So how do we come up with these general principles? And one of the things that we really need to do is speed up the process by which we get to these general principles, right? So, and I see that collaboration, FAIR principles, and modeling are some ways that we can get to these general principles. So this is sort of a roadmap for how science might translate, but I think we also need to be quite honest about how science works, and it's really not a linear array. It is a fairly meandering process, right? And so a lot of scientific threads are going to end in dead ends, and that's okay. We've learned something along the way. Some of them are going to go off onto their own little tangents, which might end, or they might have deviations that have some really new important insight that turns out to be important, that we wouldn't have planned for in the beginning. So I think we should be honest in how science works and embrace that complexity of the scientific process. So I'm going to be examining some of that meandering through the lens of comparative seascape genomics. So simply put, this is taking multiple species of corals in this case, and I'm going to be focusing on the cnidarian part of the coral solely in this talk. And we're going to be looking at this against the backdrop of the Great Barrier Reef and looking at from a genomic point of view. So I'm focusing on genomics because genomics has really amped up our power to discern patterns that we really couldn't see when we were looking at few marker genetics of old. So I'm really focusing on genomics, and you'll see that there have been some new things that come to light because we now have the power to resolve species differences, to look at adaptation, and so forth. Okay, so setting out on this research program, there were some things that I wanted to make sure that we could focus on. One of them is dispersal connectivity. So dispersal is one of the most important traits of an organism. And using genomic tools, we can do things like identify isolated and well-connected populations, estimate average dispersal distances, compare biophysical model predictions against long-term gene flow patterns. And we can use this sort of information to do things like look at where assisted gene flow might be most useful, predict larval supply that's originating from protected or restored locations. So these are just some examples of how we could use information on dispersal connectivity. The other thing we can do with genomics that's really great is we can start looking at adaptive genetic variation. And so here we can do things like find candidate genes for heat tolerance or other traits of interest. We can predict individuals and populations that are more or less adapted to future conditions. And we can identify locations with high or low genetic diversity. And we could use this information to protect future adapted populations, to use future adapted individuals as parents in coral breeding programs. We could prioritize source and recipient populations for assisted evolution, and so forth. So these are just some ideas of some things that we can use this information. Okay, so this was the plan. You know, look at multiple species of corals, and we'll make some great inferences about dispersal connectivity and adaptive gene flow. But of course, science doesn't work the way you plan. And so it quickly became clear that when you start digging into the genomics of corals, you start uncovering some really weird stuff that looks like you might be dealing with multiple taxa. And so we kind of have to deal with that before we can start looking at dispersal and adaptation. So that sort of leads off into some other directions, which is like, well, you know, how common are these cryptic taxa? Are they ecologically different from each other? Do they matter, right? So, so we meander around looking at that sort of stuff. And then we might want to look more closely at these cryptic taxa and what they're doing with each other, whether they're actually exchanging genes with each other, which then also creates some additional questions like how common and how does that lead, sorry, I skipped too fast, how does that lead into adaptive genetic variation? So this is the meandering roadmap for my talk today. It's a little bit non-linear, but I will lead you through these different paths, and we'll come out the end knowing something more about corals, I hope. All right, our backdrop is the Great Barrier Reef, which is an amazing place to be and work. In the Great Barrier Reef, coral reefs have persisted since the last glacial maximum, but the modern reef is quite young, and it is well connected to the Coral Sea. So you see these major oceanic jets coming in. The environment itself is fairly heterogeneous. There's a gradient in temperature on the far left from north to south, not surprisingly, of hot to cooler temperatures. And the other thing that I think is worth noting is that the currents are really quite complex in this seascape. Okay, and these are our star players. These are the main characters I'm going to be talking about today. And I'm just grouping them by their reproductive mode. So corals have two basic modes of reproduction. We can classify them as being broadcast spawners, where the gametes are released into the water column and the zygotes float themselves before becoming planula larvae and settling, as compared to brooders, where fertilization happens on the mother coral, and the planulae have a shorter duration. Okay, so we have a number of broadcast spawners that I'm going to show you some data on, and we have two brooders that I'm also going to show you data on. And Stylophora pistillata is going to be kind of our model species. We have more information on Stylophora, and so I'm going to give you a lot of case examples of Stylophora, and then I'm going to fill in with other species where we have that comparative data. All right, so here we go on our map of where we are headed, and I'm going to start by looking at cryptic taxa, because we need to do this before we can do other things. All right, so cryptic taxa, I'm just going to define these as genetically distinct groups that are not easily recognizable based on gross morphology. I'm calling them taxa because I'm agnostic as to whether they're actual species or not. I know different definitions are used on species. We can have some friendly conversations about that later. So let's imagine that you go out and you sample corals from two different locations, and you do your genetics and you find that they're slightly different from each other. Well, you have two hypotheses that can explain this result. One might be that you have differentiation by distance, so they're just dispersal limited, and there's gene flow is low, or you might in fact have reproductive isolation or different species that you're dealing with. You can't tell that apart if you've only sampled from two places. But if you sample in one place and you find that you have next to each other things that are genetically distinct, you can be really pretty confident that that's living together, or symmetry, is caused by reproductive isolation. So that gives you an angle, and if you see that pattern repeated, you can be really even more confident that you have at least two taxa here, the green and the blue one that we're dealing with. So our criteria for cryptic taxa that we're using is that we have genetically distinct groups and that these groups are sympatric with each other. Okay, so how do we actually apply this criterion? So we sample, in this case Stylophora, we sample a lot of individuals, we genotype them, and then that's a huge amount of data. So principal components analysis is a really easy way to visualize that data complexity. So I'm going to show you a lot of PCAs today because I think it's a really digestible way of looking at the data. So we did this with Stylophora that we had sampled across the GBR, and you can see that it falls into 5 really distinct groups. We can use k-means clustering, and we also get 5 groups. There's a little bit of geographic differentiation within the purple, but we definitely have genetically distinct groups of Stylophora. Then what about the second criterion of symmetry? The pie charts show you where those specimens were sampled from, and you can see that we're getting more than one taxon often from the very same reef location. So our second criteria of being sympatric is met in this particular instance as well. Here's another example. I'm going to show you some more examples. So that was our first example. Here's another really recent example from the group in Stereotopora hystrix, where again we have, sorry, I've advanced too far. We have five different distinct genetic groups. These groups are sympatric at the reef scale, and even within reefs at individual locations, these individuals are living side by side. If you want to hear more about this one, go to Ilya's talk later in the week. And here's another example, Acropora hyacinthus. So this is a species complex. These are now being given their own names, but they devolve into 4 groups. These 4 groups are sympatric as well, and I'll be talking about this group later on in the talk also. So I could keep going, and I just wanted to mention that there are other groups doing exactly the same thing. So Ira Cook's group has uncovered really interesting local species of Acropora kenti, and Pim Bongartz did a really lovely paper a few years ago now that was a pretty heroic effort in looking at all aspects of these different Pachyceras. So this is a really common recurring pattern. There are exceptions. Here is an example of an exception. This is Acropora spatulata, where the groups are actually geographically well behaved and constrained, and we just see geographic differentiation between the brown and the yellow groups within the GBR region. So there are exceptions. It's not everywhere. Okay, so we kept getting this pattern again and again and again, these anecdotes, and so we thought, well, okay, how common are these cryptic coral taxa, right? So how do we go from anecdotes to generality? And so we turned to the literature. We decided to do a structured literature review. And I'm just going to show you in blue dots mean that there's unequivocal evidence for sympatric taxa, and anything short of that unequivocal strong evidence is in green. And so we pulled up all the genomic studies we could find that were published to date. And all I want you to see from this is that there are a lot of big blue dots. And it turns out that those dots, that support for cryptic taxa was found in 68% of the studied corals that we surveyed. So this is a really regular occurrence in corals. This is not anything unusual. And just a shout out to another paper that was published the same year that found very similar things. So, good ideas, multiple groups get on them at the same time. Okay. So why should we care? Why do we care that there are all these cryptic taxa running around? Well, if you find cryptic taxa, a lot of the essential parameters that you might be interested in are going to change. Your species diversity has gone up, so that's great, right? But your population size and your abundance has instantly gone down per species, so that might be relevant. I'm going to focus on this third one of species niche breadths. So what happens to our species, what could happen to species niche breadths? Why should we care? So we're going to do a thought experiment first before looking at some actual data. So imagine that this is a measured performance curve of some sort across a temperature gradient. If you assume that this is one taxon, you would conclude that this taxon can survive a wide array of temperatures, right? If you then use genomics and find that these are 2 separate taxa, then you would conclude that you have that these can live in narrower niche breadths, right? And so that might be really relevant for what you're planning to do with these corals if you're restoring them, or if you're thinking about their species distribution, where they live. This could have pretty profound consequences for how you approach your study system. Okay, so is this true? Well, we were able to test this, again, with our star species, Stylophora. And so at Heron Island, shown on the top right at Heron Reef, we have 3 of these sympatric taxa living together. And so you might recall that 2024 was a pretty hot year. And here is the temperature profiles at Heron Reef through this heat wave, and in gray are the degree heating weeks. And so this was led by Zoe Mézières. And what Zoe did was she went out to the reef and she tagged coral colonies, and then she took fragments of the same colonies and brought them into a common garden aquarium-based experiment. And so those aquarium corals were subjected to reef water coming off the reef, so approximately the same temperature as what the corals were experiencing in the water. And this is pretty much the raw data of this experiment. We don't really have to do statistics to show a result. We separated them out into their taxa after the fact. So we genotyped them after the fact. But you can see, for example, that taxon 4 was the most sensitive to the heat stress, whereas taxon 1 and taxon 5 were a lot more resilient to the heat wave that was experienced. And so those differences are significant in terms of time without bleaching, survival time, how much of the fragmented colony bleached. These were all found to be significant. Because these same colonies were tagged in the water and present in aquaria, we could also go back and ask whether there's a correlation between what happens in nature and what happens in the laboratory. And indeed, there's a strong correlation between the individual genets that survived or died in the experiment and in nature. So, but more importantly, this validates the fact that these three taxa actually are behaving in really different ways through this heat stress. Okay, so for cryptic coral taxa, they're very common, 68% of nominal species. This changes our estimates of population size and other essential biodiversity parameters, and they can differ in thermal tolerance. So let me go back a second. So I think when you should ask yourself if what you're doing with corals, if you had cryptic taxa amongst your studied species, how would that change the interpretation or the intent of what you were doing? So I think that's a question that we should all ask ourselves and think about whether this is something that might matter in terms of the work that you are doing. So cryptic species are common. But now that we have a way of identifying them, we can proceed with looking at things like dispersal and adaptive variation. And so what I'm going to do now is be focusing on a single group. So for example, in Stylophora, we're going to focus on this purple group. So this is a cohesive taxon, and we're going to look at that one in more detail. Okay. So just to remind you, dividing corals up into the two main reproductive traits of broadcast spawning and brooding, this leads to the prediction that we would expect broadcast spawners to disperse further than brooders. So first, I'm going to show you a comparison between two different taxa, and we have our friend here, Stylophora, on the right, and we're going to contrast that to broadcast spawner of Poecilophora vericosa. So these two taxa, they're sampled from the same locations on the GBR, and just first showing you the PCAs, you can see that they're really, really different from each other. We have strong geographic structure. This is all within one species in the broadcast spawner, where it's a bit of a splatter. The brooder has a strong geographic structure. The broadcast spawner is more of the splatter situation. So, just looking at the raw data, it looks like they're pretty different, but we can do some proper analyses. And so we're using an isolation by distance approach to estimate dispersal. And so what we need are two pieces of information. We need the isolation by distance slope, and we need information on abundance. So to get the isolation by distance slope, we basically regress the geographic distance between coral colonies and their genetic distance, and we get this slope estimate. I'm not going to run through how we do abundance, but happy to have a chat about that if you're interested. And so from these two pieces of information, we are able to get this really important parameter called sigma, which is the generational dispersal distance in one dimension. So if we do this for the brooder, we get a sigma value that is within meters. So under 100 meters is the mean dispersal per generation of this brooder, which is not very far, clearly. We can contrast that to the broadcast spawner, and here we find dispersal on the scale of kilometers, right? So these two things are behaving really differently in the same seascape in terms of their dispersal ability. But again, you know, and then we can just visualize this on a map. If you put a hypothetical parent in the middle of the ring, you would expect 50% of the offspring to be within that red ring that I've shown here. Okay, so that gives you an idea that the broadcast spawners are really dispersing across the GBR properly, whereas the brooders, their dispersal is really within individual reefs. And so we can ask, okay, well, that's, you know, 2 species, n equals 2. That's not super convincing. It's interesting, but not convincing. So is this a general pattern? And so this is some really epic work done by Catherine Prada, where she went to the literature again, and she actually reanalyzed all the studies she could find that had the data that we could use. And so I just want to draw your attention to the blue band here. These are the brooders. And she found consistently the brooders were dispersing under 1 kilometer. And then the broadcast spawners in the orange and red colors are dispersing between 1 and 100 kilometers. So really different ways that these organisms are interacting with their landscape. If you have questions about this process, Kat's a great person to talk to and has made all sorts of resources for how you can do these analyses. And I really want to thank the people who made their data available so that we could do this sort of work. So in summary with dispersal, these different corals that have different reproductive modes are differing by an order of magnitude in their dispersal. So this is now, we can confidently say this is a general principle. We can use this rule of thumb now when we are thinking about different species and how they disperse on the landscape. Okay, so we need to think very carefully about how these different life histories are going to translate into different vulnerabilities under climate change and how we can manage them differently based on this observation. All right, so then what about adaptive genetic variation? So I'm going to look at that in 2 different ways, and we'll just make a contrast between one approach being to look at correlations between genetic patterns and environmental variables. So it's sort of regressing genotypes on environment, essentially. That's one way of looking at adaptive variation or identifying adaptive variation. Another approach is to look at total genomic diversity with the assumption that things that are more diverse have more capacity to evolve to whatever circumstances they face in the future. The conservation genetics literature is actually undecided at this point which of these approaches is more useful in the long term in a changing world. And so I'm going to show you some data that follows both of these approaches. But we'll start with the genotype environment associations. And these are good for asking questions like, are there strong candidate genes for heat tolerance or any other trait? Which environmental gradients drive local adaptation? Can we predict individuals and populations that are more or less adapted to future conditions? These are the sorts of questions we can ask. I'm only going to focus on the last one today, just in the interest of time, and just show you an example of one of the ways we can use this. So to forecast how well populations are adapted to future conditions, this is a little bit of a recipe. There are a lot of different methods, but in essence, they boil down to a similar sort of approach. The idea is that you build a statistical model where you measure environmental variables, you look at your genotypes, and you come up with a predictive model of those genotypes based on environmental variables. You then take future environment, and then you compare that, you use your statistical model that you've built to come up with the genotypes that would be well suited to that future environmental situation, okay? And then the final step is to basically look at the difference between the genotypes that are needed in the future and the genotypes you have now at a particular location. And so we're assuming there's no dispersal. We're assuming a lot of other things that we maybe won't talk about right now. But this is the sort of thing that you can come up with as a prediction, where you can look and see a metric of how well populations are predicted to be adapted to future conditions or misadapted to maladapted to those future conditions. So not surprisingly, on the right, under the more extreme climate scenarios, we have populations that are predicted to be much more maladaptive, whereas more moderate or middle of the road scenarios, we have a mix of reefs. So reef situations. So I think what's surprising here is that even under this moderate middle of the road scenario, under these predictions, there are some reef locations that are predicted to be pretty well adapted to those future conditions. So that's some good news. What's also surprising to me, at least, is that we get this mix of sort of good and bad populations. It's not a simple longitudinal gradient. We get good and bad reefs both in the north, that's going to be hotter overall, and in the southern GBR. And so if you want more details and a deep look at the genes and loci that might be part of this story, Zoe's going to be giving a talk on this later this week. So there's a major caveat in this sort of approach, which is we don't have experimental validations of these predictions. These are predictions only. We don't have experimental validations to date. What I can say, though, is work that is ongoing in plants systems has shown that these sorts of approaches do a really good job of predicting. They do quite a good job of capturing relevant predictions about which populations and which individuals can respond to future conditions. And what's really nice is that this approach is fairly inexpensive and scalable. So you could imagine doing this for a lot of different species because you don't have to go and measure phenotypes for every individual. We're letting the environment tell us about the phenotypes. Okay, a contrasting approach is to look at total genetic diversity, where the assumption is that this total genetic diversity should tell us what a population's overall capacity for adaptation is, or its evolutionary resilience. And we can ask questions like, do we see differences amongst coral species? Does genetic diversity differ by location? And can we predict genetic diversity from spatial features? So can we extrapolate? I'm going to focus on these. I'm going to show you data on these first two because this is also really new results coming out. So this is work that Ilya Byrne has been leading. And so what we did was we took data that we have, whole genome sequencing data from these 5 different species, and I'm just graphing for you a measure of genetic diversity that's commonly used. This is coverage-corrected data. If you want to have a chat about that, we can talk about it. But one of the things, so one thing you can observe straight away is each one of these species is showing a different level of genetic diversity. So that's not very surprising, but again, it means different species have different attributes. We shouldn't be just lumping them all together. But the other thing that I think is quite interesting is if you look at these lower 2, you will notice that these blue populations, which are the Capricorn bunker group, have much less diversity, and also Pelorus Island has less diversity than some of the other locations in the GBR. Some of you undoubtedly have observed that the top 3 are broadcast spawners, and the bottom ones are brooders. And so this might be a difference between these 2 groups, but we also have a strong case of taxonomic non-independence here. So I'm going to take that as a possible pattern that is emerging. So if we look at adaptive variation, just to summarize, I think the initial results in using these genotype environment approaches are promising to identify future adapted populations and individuals, but experimental validation is needed. And we need to look at symbionts too. And Hisatake Ishida is giving a great talk, I hope, on Friday that is looking at the symbiont side of these same corals that I've been telling you about today. So if you're interested in symbionts, you can see the other side on Friday. In terms of total genetic diversity, coral species differ clearly in their total diversity. We find spatial homogeneity within the broadcast spawners and heterogeneity within the GBR brooders. And there's a whole group here giving a variety of talks that's really interested in how do we predict that spatial diversity. And so I encourage you to catch some Climate Reef talks, and Helen Fox is giving an overview on that. If you're more interested more generally in what we're doing at Ames and how adaptive genetic variation can be used, Lena Bay will be giving a talk later today, so you can catch that later today. So overall, adaptation is something I think we need to build into our management strategies, and we need to know where our corals are adapted, where are those adapted populations, where are those adapted individuals. And so these are promising approaches to bring us to that state. All right, and so now in this final part of the talk, I'm going to take you off down on the left-hand side, where we're going to look a little bit more closely at cryptic taxa and look particularly at what's happening between them. So when you have when you have taxa that are reproductively isolated, they're usually called good species, which you can guess maybe what the other contrast to that is, but we'll call them porous species. So these are species that have some degree of reproductive isolation, but they're also exchanging some genetic material as well. And so you might see words like gene flow between species, hybridization, admixture, introgression, et cetera, describing that state of being. And this is an idea that's been around for a long time. Charlie Barron's book cover I've always found intriguing, and there have been some really nice review papers that have also looked at hybridization. So this idea has been around for a long time, but with genomics, we can really get in and take a look. Now, if you have early generation hybrids, this is really quite easy to diagnose. This is some nice work from the Caribbean, and you can clearly see that you have intermediate individuals that look like F1s in this situation. But if you have divergent taxa and there's really small amounts of gene flow that's been going on for a long period of time, it's a lot harder to tell apart. And so there are two basic models that we use. One is a no gene flow model, where the concept is there's an ancestral species. It splits apart into two separate descendant species. And so this is like one model of speciation, and we contrast this to a gene flow model of speciation where you have divergence, but there's connections via gene flow. And so essentially what we do is we derive theory or simulations, and then we look at our empirical data against that theory and simulations and ask whether our empirical data are a better match to these expectations with or without gene flow. Okay, so coming back to Stylophora, if you recall, we had 5 taxa. We compared every single one of these taxon pairs against these gene flow and no gene flow models. And in all cases, we found that the gene flow model was the best predictor. It's like, okay, so that's interesting. But what happens when we go back to the literature? What happens when we survey a whole bunch of different taxa? And so I know this has come out fuzzy, which I apologize for, but what I want you to see is that there is a wide variety of species. We surveyed all the studies that had used some of these different methods to test these different models. And if you see the gene flow column, you'll see it's all yeses. Every single case that tested found that their particular cryptic taxa were linked by gene flow. So this is not just a curiosity, this is a regular feature of coral biology. And it gets weirder. We have some recent work in the Acropora group. So I'm going to show you a little bit of just a taste. This is really recent stuff, where we have these 3 species that are hybridizing with each other. If you imagine on a PCA, if you put 3 species on a PCA, you get them falling into distinct regions of a PCA. But if you were to imagine that there's interbreeding between some of these parental species, then you would get intermediate forms within the PCA space. But if you imagine now three-way hybridization, you would think that the PCA, this would pull to the center of the PCA. And this is actually exactly what we find with this group, that we find these admixed or introgressed individuals pulling towards the center. This is not missing data, and this has been verified with D statistics. So we get three acroporas that are table corals that are hybridizing with each other. Three-way hybridization is very rare in nature. This is the first instance in corals that has been reported. So that's super interesting. And what's even more curious that these hybrids are found only in the southern part of the GBR. So something weird is going on here. It's super cool, and I'm gonna skip the next few slides just because I think I'm running short on time. But if you want to talk to me about the genomics of what's going on, I would love to have a chat with you. So I'm just gonna skip across those and summarize what we know about porous species, which is that coral species are clearly often exchanging genetic material. This is not just a one or two anecdotes. This is a general pattern. We should expect our corals to be playing hanky-panky with each other every now and again. Okay. In the Acropora hyacinthus complex, we have three-way admixture. It's restricted to the GBR, and I didn't have a chance to tell you about it, but we think that we have a special case of introgression, which might support rapid adaptation, which would be super cool because this would be a new mechanism that we didn't know about, whereby corals might adapt quickly to rapidly changing environments. So in this last 40 minutes or so, I've taken you on this meandering roll through some of the work that we've done. And this is how science works, is you start off in one direction and you end up going in another direction. I didn't think that cryptic taxa would be such a thing, but there we go. And I think a lot of other groups have discovered the same as they've gotten into genomics. But we've learned a lot of stuff along the way. We've learned that these cryptic taxa are super common. We've learned that they're ecologically different from each other. We've learned that broadcasters disperse further than brooders, that we can use adaptive variation as a predictive tool, that we might have interactions between spatial patterns and reproductive mode, and that porous taxa are very common, and this might be a route for adaptation. Okay, so if you've been interested in stuff I've been talking about, Thursday and Friday are going to be really busy days. There's some really cool symposia happening. And then just to summarize, because I know in a talk like this, you're only going to remember a couple of things. So here's really what I want you to remember, is the general principles that we've been able to derive from this process. So named species are unlikely to be cohesive taxa, that porous species are the rule, and that broadcast spawning and brooding corals differ in their dispersal. Those are the general things I want you to remember. I also want you to collaborate, and I've been so gratified to hear people talking about collaboration at this conference. It's really great. And I think this is something that we can really learn from traditional knowledge systems of focusing on interactions and relationships and not each other. And so please publish your data, make your tools available, and thank you to all my collaborators.
Fantastic presentation. Unfortunately, we don't have time for any questions. Cynthia, just come back though, please. So just going to present a small little thank you on behalf of you. Remember, Cynthia and team is here. So if you could just join me again for a round of applause.